that trouble. So thank you, Amy. I will just give a brief introduction and then I will go straight into the presentation on long COVID. So just here is just a brief view and notes on Stellenbosch University. The university is situated in a town called Stellenbosch, which is near Cape Town and in the heart of the winelands with some of the best wines in the world. So this is also an invitation for you to come and visit and discuss science over a glass of Stellenbosch winelands wine. I'm a physiologist in the Faculty of Science with special interest in blood clotting and the identification of novel circulating inflammatory biomarkers that may be used in the early identification of various diseases. Recently, my focus has also turned to COVID-19 and lately also long COVID. I would just like to acknowledge that the work that is presented here was done with both clinical as well as research collaborators, including Professor Douglas Kell from the University of Liverpool, Dr. Timothy Roberts, which is a statistician at UCL, and then clinical co-workers from South Africa, Mediclinic, which include hematologists, and then also biochemists from our proteomics lab, and then obviously also my technical assistant that helps a lot in the laboratory. So over the years, my lab and our collaborators from various labs over the world have been following a systems biology approach where we study circulating biomarker profiles and we focus our research endeavors on studying the effects of increased circulating biomarkers and how they interact with cells of the hematological system. We focus in particular on platelets as well as erythrocytes or red blood cells, as well as on the clotting protein fibrinogen. And we are also interested in identifying novel inflammatory biomarkers that may play a role in the development of neurodegenerative diseases and also other inflammatory conditions. Techniques that my laboratory use include ELISAs and multiplex analysis, as well as morphological readouts from super resolution and electron microscopy, as well as confocal and fluorescence microscopy. We are also interested in the viscoelastic readouts, and we use thromboelastic analysis to study blood clotting, as well as proteomics, where we focus specifically on clotting proteins. I furthermore develop and have patented novel biosensors that detect biomarker concentrations in blood samples. And these sensors will eventually be used for the early identification of disease risk. Over the years, we have published numerous papers that show the importance of inflammatory biomarkers in the development of various conditions. And here's just a few of those examples. As I mentioned, we are interested in platelet signaling and their role in abnormal clotting. As you can see from this diagram that we recently published in Seminars on Thrombosis and Hemostasis, platelets have very many receptors where circulating inflammatory biomarkers may attach to. The result is a hyperactivated platelet that can aggregate to other platelets and also attach to red blood cells as well as to endothelial cells. Platelets are therefore important signaling entities, not only in cardiovascular diseases, but also during viral and bacterial infections. There's also complex relationships between receptors on platelets and endothelial cells, where circulating biomarkers may bind to. As we all know, damaged endothelial cells are also hallmarks of cardiovascular disease. We are also particularly interested in P-selectin, a biomarker inside platelets and endothelial cells. But the P-selectin can also act as an adhesion receptor on the platelet membrane or as a soluble biomarker in circulation. So the clotting protein fibrinogen is also one of the molecules we have studied quite often. And we looked at the importance that it plays in clotting and also pathological clotting. And as you can see from this diagram, it can also act as circulating biomarker that can bind to various receptors on platelets. Lately, the importance of damaged endothelial cells, platelet hyperactivation, as well as aberrant clotting has now been well recognized in COVID-19. So we are particularly interested in the structural changes in the biochemistry of the fibrinogen molecule and how circulating inflammatory biomarkers may directly bind to this clotting protein to induce protein misfolding. If you focus your attention to the two cartoons shown in figure A, On the left is a healthy protein structure where you will find many alpha coils and a few beta sheets. However, in the presence of inflammation or oxidative stress and various inflammatory circulating biomarkers, this structure changes, where the alpha coils will untwist into beta sheets, as seen in the cartoon A on the right. If we look at various individuals with when they are healthy and we look at their clot structure, you will see that there's a fine net that form, and these nets mainly consist of alpha coils, and the healthy clot you can see in figure B. In various inflammatory conditions, including cardiovascular disease and diabetes, we see a clot structure with a similar structure seen in C, where the clot forms a denser matted, has a denser matted appearance. And this obviously makes for the aberrant clotting and contributes to hyperclotting and hypercoagulability in these type of conditions. So to our knowledge that we've gained from the research we've done on platelets and abnormal plasma protein structure in various inflammatory and cardiovascular diseases last year, we focused our attention to COVID-19. So in early 2020, we published a review to discuss our take on clotting and endothelial platelet and erythrocyte dysfunction in COVID-19. And we argue and reviewed literature that point out that, firstly, vascular implications of COVID-19 may result in clotting protein and circulating biomarker endothelial, as well as red blood cell and platelet dysfunction. And we reviewed the various biochemical processes and pathways associated with vascular dysfunction. And we focused particularly on fibrinogen and D-dimer formation, as well as our biomarker of interest P-selectin, as well as von Willebrand factor. And we concluded in the review that probably the best option for these conditions in the acute phase would be to use point-of-care devices as part of a clinical analysis, and methodologies that uses point-of-care devices that might focus on thromboelastography and aggregometry as platelet dysfunction, as well as fibrinogen and clotting dysfunction seems to be a major culprit in these patients. So as shown in diagram A, a typical pathology in bleeding and clotting, we will see a seesaw balancing act between bleeding and thrombocytopenia and hypercoagulation, with a healthy clotting in the center. In diagram B, we depicted the process of disease progression as seen in the clinic by our clinical collaborators, where early in the disease, the patients present with clinical manifestations of hypercoagulation, but unfortunately, if left untreated, thrombocytopenia and bleeding can result in the later stages of the diseases. And then obviously follow, the cytokine storm will follow with dire consequences for the patients. In our view, therefore, quite early in the disease, early 2020, we thought the best chance of survival would be to treat the patient that lands in the ICU during the green block in the B cartoon, which is the optimal time of intervention that will ensure the best outcomes for the patient. So in order for the patient's best survival, to treat abnormal clotting and not wait until bleeding or thrombocytopenia will happen. So during the abnormal clotting phase of the disease, fibrinogen levels are high and D-dimer are still normal to slightly increased, but increasing, where in the later stages of the disease, fibrinogen may be depleted with an increased level of D-dimers in circulation. And as just mentioned, if the disease is left untreated, bleeding and cytokine storm will ultimately cause thrombocytopenia and push the patient into a situation where the disease can probably not be turned around. So we also looked closely at fibrinogen and structural changes, and it was from the data that we gathered from patients in the hospital. We think that, or we saw that as the COVID severity increased, more D-dimer were present and fewer soluble fibrinogen molecules seemed to be present in the blood samples. Now, with this in mind, we looked at platelet-poor plasma of patients and exposed it to our fluorescent marker theoflavin T. And theoflavin T binds to open hydrophobic areas in proteins and have been used quite often in the past to identify amyloid protein in brains of Alzheimer's disease patients. So a few years back, myself and Doug Descal discovered that it also marks misfolded fibrinogen and fibrin proteins during pathological clotting. And in this figure, you can see we looked at platelet-poor plasma protein structure in COVID patients, and we compared it to healthy and diabetic patient plasma samples. So this simply was whole blood, citrated whole blood, centrifuged with platelet-poor plasma prepared and with added theoflavin T. So no thrombin added, just simply the plasma with the theoflavin T. So if we look at control plasma smears, we found that there are small areas of nearly undetectable abnormal clotlets that would form spontaneously in plasma of such healthy individuals. But more abnormal clotlets were noted in plasma of individuals with diabetes. Now, previously we published quite extensively when we looked at plasma protein structure, but with the addition of thrombin to cause major extensive fibrin clots, where the soluble fibrinogen was changed to insoluble fibrin fiber nets, but in the presence of thrombin. So what was quite significant to us was the extent of the abnormal clotlets that we found in acute COVID-19 plasma that you could see in the samples shown in the right micrographs. So this was quite interesting. If you look at that size of those abnormal clotlets, you could really think that they might block small vessels in the lungs and cause severe damage to lung tissue in particular. So we were also interested in the structure of platelets and the interaction with erythrocytes in these individuals. And platelets, as I mentioned, have receptors for fibrinogen, and also the spontaneous form fibrin clotlets can bind to these receptors that they have and cause hypercoagulation. So it was quite interesting for us to look at how platelets and red blood cells interact in COVID patients. So in the figure on the left, you could see platelet aggregation and shedding of the content and just aggregating to each other and just looking quite sad, I must say. When we compare then red blood cell interactions, we see that platelets and red blood cells form quite extensive interactions where the platelets attach to the red blood cell membranes. So the question arose was, what we see here in the patient blood, was it actually something to do with the spike protein that we see all of these changes to the fibrin, to the platelets, and to the red blood cells? So we took spike protein, 1 nanogram per milliliter, which is quite a low concentration of spike protein S1 of the South African variant. And we added it to fluorescent purified fibrinogen followed by thrombin to create an extensive net. So in A, you could see just the fibrinogen, purified fibrinogen with thrombin without anything added to it. And then in B, we added this spike protein to it, where you could see immediately lots of gunky deposits can be seen with significant abnormal clots forming in the clots of purified fibrinogen. We also then added spike protein to platelet-poor plasma of healthy individuals, and you could see that in A and in B. And we also created extensive clots in C and D by adding thrombin. So the important thing that we noted was spontaneous clotlets in areas of anomalous or amyloid clotting in plasma proteins are also seen when we add theoflavin T to these samples. So we redid the experiment with healthy individuals, and we looked under the scanning and electron microscope. And in A and B, you could see red blood cells of a healthy individual with B little platelets putting out little pseudopodia when it attaches to the glass surface where we deposited the sample on, it's whole blood. And with spike protein, we immediately saw platelet hyperactivation. You could see severe spreading, for example, in D, but also what was quite interesting to us is we see these spontaneous clotlet formation And some of these areas you could see in G, which is similar to the ones that we saw in our fluorescence microscopy micrographs. So definitely spike protein triggers something in the blood that causes abnormal changes to both the fibrinogen, the platelets, and then red blood cell platelet fibrinogen interactions too. So the next step that we wanted to look at is our experiment looked at proteomics of healthy plasma with and without spike protein. And this was initially only done with 4 individuals, but we now repeated it with up to 10 individuals. So we prepared platelet poor plasma from citrated blood, and then we added trypsin to the sample, to the platelet poor plasma, for the degradation process and for proteomics. To our surprise was we found that in the Eppendorf's with the spike protein added to healthy individuals' plasma, there was a significant visible deposit at the bottom of the tube that you could actually see with your bare eye. So the supernatant was run through the mass spec, but after a C18 filtering step to try to filter out some of the, if there's anything left. And so what we then discovered here was that even with trypsin degradation, the lytic process is severely affected when spike protein is inside the plasma of a healthy individual. So our collaborator at the proteomics lab analyzed 1 microgram of protein of the supernatant per sample. And what we found was structural changes to the beta as well as the gamma fibrinogen chain, complement 3 and prothrombin. And these proteins were substantially resistant to trypsinization in the presence of spike protein is 1. Here we also suggest that in part the presence of the spike protein in circulation may contribute to the hypercoagulation length that we see in COVID-19 positive patients, and which may cause substantial impairment of fibrinolysis. So the variation of the protein concentrations between the naive sample and when spike protein is added, we believe point to the fact that these molecules may actually be trapped in the deposit that we actually could visualize in the Eppendorf tubes, and that it may play a significant role in the hyperfibrinolysis process. So with this information in mind, we turned our attention to LongCovid. And as you all know, recently researchers has said that about 25 to 35% of individuals that have recovered from acute COVID-19 still suffer from various complaints like shortness of breath and heart palpitations and chest pains, sometimes even to such an extent that many of these individuals cannot work and fulfill their daily tasks. So we decided to repeat the experiment with a plasma of controls, healthy individuals, as well as type 2 diabetes patients and acute COVID-19 patients and long COVID patients. So we decided to repeat the experiments, particularly with diabetes patients as well, as we know that type 2 diabetes patient plasma reacts quite or patients with type 2 diabetes react quite badly when they get COVID-19. So we wanted to see if it's a phenomenon that this deposits that we see is perhaps not only in COVID plasma or in long COVID plasma, but perhaps also in conditions like type 2 diabetes. And we wanted to see if we could find the link between these 3 types of diseases and healthy individuals. So the experiment that we did for proteomics, we looked at healthy plasma, type 2 diabetes, acute COVID and long COVID. And we added the same procedure. We added in a degradation step with trypsin. And then we looked at hypercoagulation in these samples. So when we looked at the experiment, to our surprise, we found that there was a visible deposit at the bottom of the Eppendorf of the acute COVID. and of the long COVID samples, but not in the diabetic sample, and also obviously not in the control samples, and that we already showed previously. So this suggested that trypsin could degrade all of, at least most of the plasma proteins that is present in healthy plasma and in the diabetic sample, but not in the sample that contains non-COVID plasma or acute COVID-19 plasma. So for proteomics, the sample supernatant was again filtered with C18 filter, but we then studied the unfiltered in our laboratory with fluorescence microscopy. We studied unfiltered, so we didn't use the C18 filter, supernatant with our fluorescence microscope. So our collaborator in the proteomics lab analyzed the 1 microgram protein of the supernatant, as well as 1 microgram of the gunky deposits at the bottom per sample, which he again used trypsin step, but he treated it with other chemicals to get the deposit at the bottom soluble. So what we found was, if we looked at structural changes in plasma after trypsin degradation in the supernatant, in healthy plasma as well as type 2 diabetes plasma, after trypsin digestion, there was literally nothing visible in, or very little visible in the sample. However, in this supernatant, unfiltered supernatant in COVID-19 plasma, after trypsin digestion, we could just still see these massive deposits of platelets. And if you just turn your attention to the scale bar, 10 micrometer, it's quite extensive deposits present that is not taken or digested in the COVID plasma sample. The structural changes in the Eve plasma in long COVID samples without trypsin was also quite interesting for us. Luckily for us, we had stored plasma sample of a healthy individual before they actually contracted COVID-19. And we had his sample when he was complaining from long COVID. So you can see we really had to search quite long and hard in his sample to find this little 2 dots that we see in A in a tile scan. But in B, you can see his sample that still contain, this is obviously no trypsin, these big deposits. And in C, you could see another long COVID sample. And this we saw in all of the long COVID samples, that they still contain these deposits that even after they have been fully recovered from acute COVID. So all of these samples were also treated with the theoflavin T. So structural changes in plasma in long COVID after trypsin digestion is shown here, and without filtering. And you could see the trypsin did not really do anything to these deposits. They're still present in the plasma. So I just want to remind you again of the clotting in the naive plasma of COVID, and compared to long COVID naive sample where trypsin was added. So no trypsin, and trypsin didn't really matter. It seems as if degradation doesn't do anything to the clotted deposits that is still contained in the plasma. So this led us to file a patent just recently, 2 or 3 weeks ago, that would help us to diagnose long COVID using the detection of these large deposits before and after enzymatic lytic methods. So, and we also believe that the invention could perhaps assist with a diagnostic tool for a point of care test to identify long COVID as a clotting disorder in patients. So the proteomics analysis is still really very new. It is just out yesterday, so I'm not going to say too much about it. But as mentioned, we also obviously looked then at the proteomics of the supernatant, but we also looked at the undissolved, unlysed pellet. We treated it, and we could eventually trypsinize it so that it could be put through a mass spec. And what is interesting for us is if you look at these diagrams, if we compare the controls, obviously there is not a clot. The controls didn't have a clot at the bottom, but we just took the same volume that was left in the tube that contained in the COVID and the long COVID sample, the actual deposit that you could visually see. And we looked at which proteins are similar and overlap and which are totally just present in the long COVID versus the COVID clots. So at a first glance, it seemed to us that there are 5 main protein changes in the deposits after trypsin digestion. And this is now the deposits that formed that we again digested with trypsin. And serum amyloid A seemed to be one of the molecules that is prominently visible in this proteomics analysis. Obviously fibrinogen, we would expect that, and then some of the complements, and also cytoskeletal keratin. These are the molecules that were present in the long COVID sample, but not in the other samples. So this was quite interesting for us because we think cytoskeletal keratin might be, while that is present, might show or point to damage of the muscle in the small little vessels, the blood vessels. And then obviously serum amyloid A is an acute phase molecule. And there have been a lot of publications recently that show that serum amyloid A is also really an important marker in acute COVID. So it's very interesting that it seems as if these deposits keep on and stay in the plasma in some individuals, that will then result in them having an abnormal clotting profile when they have long COVID. So the treatment options was now quite interesting. So obviously we work with our clinical collaborators, which see patients all the time. And we found, or they found, that they have great success when they treat individuals that come to them with long COVID. If they treat them with anticoagulants and molecules or products or medicine that reduce platelet activation. And even 2 days after these long COVID patients have been given this option of treatment with anti-clotting agents, they seem to have a good reaction to the medication, where they don't have the shortness of breath anymore and the lung issues. So obviously this is something quite new, and this is only in 10 patients. What was interesting, our clinical collaborator who puts the patients on these regimes when they have them in hospital, when they early on in the disease treat them with anticoagulants and anti-clotting agents, it's not those patients that seem to come back with the long COVID issues. It is the patients that have not been treated when they've had acute COVID with the anticoagulant molecules and medicine. So that is the interesting thing that I would like to share with you. We are currently preparing the paper with the long COVID data, and I will share it with Amy as soon as we have that. Thank you very much.